We're still on chapter 25, and now we are going to talk about nephrons. And nephrons are the structural and functional units that actually form urine. And there are more than 1 million per kidney, and there are two main parts. There's the renal corpuscle and the renal tubule. There are two parts of the renal corpuscle. There's the glomerulus, which is just a tuft of capillaries composed of fenestrated endothelium, highly porous capillaries, and what they're going to do is allow for efficient filtrate formation. So we actually want these capillaries to be porous because we want stuff to leave the capillaries to form this filtrate. And the filtrate is a plasma-derived fluid that the renal tubules will then process in order to form urine. The other part is the glomerular capsule, or the Bowman's capsule. And this is a cup-shaped hollow structure that surrounds the glomerulus. And there's two layers. There's the parietal layer and the visceral layer. The parietal layer is composed of simple squamous epithelium. It's on the outside, and it doesn't play a role in filtration. The visceral layer actually clings to the glomerular capillaries, and there are these branching epithelial cells called podocytes. And they have these extensions that terminate in foot processes that cling to the basement membrane. Between the foot processes are these things called filtration slits that are going to allow filtration to pass into the capsular space. So let's blow this up a little bit. So here we have a renal corpuscle, a glomerular capsule, and the glomerulus. The glomerulus is the ball of capillaries inside, and the glomerular or Bowman's capsule is this cup-shaped thing on the outside. When looking at the glomerular capsule, there's the parietal layer, and remember this is composed of simple squamous epithelium, and this is not going to play a role in filtration. The visceral layer has these podocytes, these branching epithelial cells with these foot processes. And these little between these little foot processes, we find the slits. And this is going to allow filtrate to pass through. Now we move on to talking about the renal tubule and collecting duct. The renal tubule is about 3 centimeters or 1.2 inches long. And it consists of a single layer of epithelial cells, but each region has its own unique histology and function. And the histology and function are important to learn. And there are 3 major parts: the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule. The proximal convoluted tubule is closest to the renal corpuscle, and then it moves into the nephron loop, and then it moves into the distal convoluted tubule, which is going to be furthest from the renal corpuscle. And then the distal convoluted tubule is going to drain into the collecting duct. So we're going to start off by talking about the proximal convoluted tubules. And these are the proximal convoluted tubule is composed of cuboidal cells with a lot of microvilli. And these microvilli are going to increase the surface area. And that's going to be really important because the proximal convoluted tubule plays a really important role in reabsorption and secretion both. And then they also have large mitochondria, and this is because mitochondria make ATP, and there's a lot of active transport going on. And in order for active transport to occur, ATP is necessary. And then the proximal convoluted tubule is confined to the cortex. So we see all the microvilli, which are going to increase the surface area, and then the mitochondria, the large mitochondria. Next is the nephron loop. It used to be called the loop of Henle. It's a U-shaped structure that consists of 2 limbs. The descending limb, this is going to be the first part, and the proximal part of the descending limb is continuous with the proximal tubule. And the distal portion is also called the descending thin limb. And then there's the ascending limb, and it's a thick ascending limb. It's actually thin in some nephrons. And then this is an image of the cells of the nephron loop. And then we have the distal convoluted tubule. This is going to be composed of cuboidal cells. They don't have as many microvilli, and they have more of a function in secretion than they do reabsorption, and they are also confined to the cortex. And so we have, again, cuboidal cells, very few microvilli. And what we have here is a glomerulus, that ball of capillaries. We have the glomerular capsular space on the outside. And this is going to be kind of hard to point to, but where the tip of these fingers on this little hand are is pointing to the parietal layer of the glomerular capsule, which would be composed of simple squamous epithelium. And then we have a proximal convoluted tubule here, and we can tell that it's a proximal convoluted tubule because it's very fuzzy, because of the fact that there's so many long microvilli. And then we have the distal convoluted tubule, which is clear, and that's because there are very few microvilli. Now we move on to talking about the collecting ducts. And there are two types of cells. There are the principal cells. They have short microvilli, and they're going to maintain water and sodium balance. And then there's intercalated cells. These are cuboidal cells with abundant microvilli, and they're going to help maintain acid-base balance of the blood. The collecting ducts receive filtrate from many nephrons. I'm going to show you what that looks like here in a second. And they run through the medullary pyramids and give the pyramids their striped appearance. The ducts will then fuse together, and it's going to deliver the urine through the papillae into the minor calyces. So we have the two types of cells. We have the principal cell, very few microvilli, and it's going to play a role in water and sodium balance. And then the intercalated cell, which does have microvilli, which is going to play a role in acid-base balance. And then this is the whole nephron. And we can look at it like this, which is going to be helpful. So again, glomerulus, glomerular capsule, proximal convoluted tubule, which is going to be composed of those cuboidal cells with lots of microvilli, going down to the descending limb, and then it's going to go back up the ascending limb to the distal convoluted tubule, which is also composed of cuboidal cells, but there are not as many microvilli. And then this is going to go to the collecting duct. You can see the collecting duct has all these branches coming off of them, or coming off of it. And each one of these branches would have a distal convoluted tubule attached to it. So it's actually going to be collecting urine from multiple nephrons. There are two major groups of nephrons. The cortical nephrons make up about 85% of nephrons. And these are the ones that are typically functioning. They're just going to make your run-of-the-mill urine. They're almost entirely in the cortex, probably why they're called cortical nephrons. And then there's the juxtamedullary nephrons. These have long nephron loops that go deep into the medulla. The ascending limbs have thick and thin segments, and what they're going to do is they're going to play a role in producing concentrated urine. So let's look at this. Again, we would have the glomerulus here, proximal convoluted tubule coming off. This is, by the way, this is a cortical nephron. It has a short loop, and it's almost entirely in the cortex. And this is just going to make the typical urine. 85% of the nephrons are cortical nephrons. Then we have the juxtamedullary nephrons, I should say. And I'm going to actually back up because it's so long. And you can see it has this long loop, long nephron loop that's going to go deep into the medulla. And this is going to play a role in producing concentrated urine. I also want to point out these capillaries, because I believe on the next slide we're going to talk about these capillaries. So around the cortical nephron, we have these things called peritubular capillaries. And around the juxtamedullary nephron, we have, around the loop anyway, we have the vasa recta. And that's what I just said. So the renal tubules are associated with two capillary beds, the glomerulus and the peritubular capillaries. And then the juxtamedullary nephrons are associated with the vasa recta. The glomerulus is a ball of capillaries that is specialized for filtration. And it differs from other capillary beds because of the fact that it's fed and drained by an arteriole. So if you think back to the chapter where we talked about blood vessels, typically a capillary bed is fed by an arteriole, and then at the end of the capillary bed, the blood, everything continues onto a venule. So with the glomerulus, we're actually feeding and draining by an arteriole. The afferent arteriole enters the glomerulus and it's going to leave via the efferent arteriole. The afferent arteriole arises from cortical radiate arteries, and the efferent is going to feed into either the peritubular capillaries or the vasa recta. And that's going to depend again upon whether or not we're talking about a cortical nephron or a juxtamedullary nephron. If it's a cortical nephron, then it's going to feed into peritubular capillaries. If it's a juxtamedullary nephron, it's going to feed into vasa recta. The blood pressure in the glomerulus is high because the afferent arterioles are larger in diameter than the efferent arterioles, and arterioles are high resistance vessels. The peritubular capillaries are low pressure porous capillaries that are adapted for absorption of water and solutes. And again, they arise from the efferent arterioles and they cling to adjacent renal tubules in the cortex and they empty into venules. The vasa recta, long, thin-walled vessels that are parallel to long nephron loops of juxtamedullary nephrons. They arise from efferent arterioles serving juxtamedullary nephrons, and this is instead of peritubular capillaries. So we don't have both. It's either one or the other, and they're going to function in the formation of concentrated urine, which makes sense because the juxtamedullary nephrons play a role in the formation of concentrated urine, so the vasa recta also do the same thing. We have a glomerulus here, and then a wider afferent arteriole, and this more narrow efferent arteriole. And this is just showing everything once again. So, cursor, there we go. So again, these cortical nephrons are almost entirely in the cortex, and they have the peritubular capillaries associated with them. So basically, we would have the afferent arteriole coming in, glomerulus, efferent arteriole coming out, going to the peritubular capillaries. For the juxtamedullary nephron, we would have the afferent arteriole coming into the glomerulus, leaving via the efferent, going to the vasa recta. Now we're going to talk about the juxtaglomerular complex, or the JGC. Each nephron has one juxtaglomerular complex, and it involves modified portions of the distal portion of the ascending limb of the nephron loop, and the afferent, sometimes efferent arteriole. It's important in regulating the rate of filtrate formation and blood pressure. And there are three types of cell populations seen in the juxtaglomerular complex. The macula densa are tall, closely packed cells of the ascending limb. And they're going to contain chemoreceptors that sense salt or sodium chloride content of the filtrate. Then we have the granular cells, or JG cells, and these are enlarged smooth muscle cells of the arteriole, and they act as mechanoreceptors to sense blood pressure in the afferent arteriole. They contain secretory granules that contain the enzyme renin. And then finally, we have the extraglomerular mesangial cells, and these are located between the arterial and tubule cells. They're interconnected with gap junctions, and they may pass signals between the macula densa and granule cells. So first, let's look at the macula densa here. Again, part of the ascending limb of the nephron loop, and it's going to sense the sodium chloride concentration inside the filtrate, which would be inside this tube. Then we have the granular cells are here. Granular cells are here. These are the smooth muscle cells. And then we have and these are going to secrete renin. And then we have the extraglomerular mesangial cells, which may pass signals back and forth between these two cell types. These are the questions for this segment. And that's all I have for you for this talk.